Scientists working with the nematode Prostonachus pacificus introduced a mutation in a single amino acid that destroyed this worm's ability to distinguish between what worms they considered food and what worms they considered kin, essentially creating a cannibal worm. This worm typically feeds on other nematodes such as the species C. elegans and bacteria. Scientists named this protein Self1 because it appears to be involved in the nematode's ability to, well, recognize itself. Self1 is a small protein of only 63 amino acids. Researchers look to see if there are genes in other species that are similar to the Self1 gene, and they found only similar DNA sequences in Prostonachus species. Because this gene is restricted to the Prostonachus genus, this led researchers to two hypotheses. One, that the gene encoding Self1 arose de novo from a non-genic region, so a region of DNA that does not contain any genes. Or two, that perhaps the Self1 gene arose from a very rapidly evolving gene that no longer bears much resemblance to the gene it was before. So in summary, this small protein, Self1, allows this species of worm to focus on what worms it considers food and what worms it considers family. Okay, so back to this concept of self-recognition. This sounds very similar to what the immune system does. This concept of recognizing self versus non-self. Where does this come from? One ancient mechanism, essentially the identification and destruction of foreign DNA in bacteria, is called the restriction modification system, or RM. RM involves the destruction of foreign DNA by enzymes called nucleases. It also involves the identification and flagging of its own DNA as self so that it doesn't come by and destroy its own DNA. The bacteria's method of flagging its own DNA as self is through the addition of methyl groups onto its DNA, a process called DNA methylation. So when this foreign viral DNA enters into the bacterium, it will not contain methyl groups on it, and it will be destroyed by the bacterium's nucleases. There are many different types of RM systems that have evolved that have differences in both the enzyme that does the methylation or the nucleases. But what's crazy to me is that the DNA that encodes for this RM system, so the gene that gives rise to the nuclease that will chop up the foreign DNA, or the gene giving rise to the enzyme that does the methylation, these are in and of themselves foreign DNA. Wait, what? Okay, so bacteria frequently transfer DNA between each other in what is called horizontal transfer. We typically think of sharing DNA linearly through what we call vertical transfer, so from DNA getting passed down from parent to offspring. So this horizontal transfer would be like me giving you a chunk of my DNA. So considering that researchers think that a lot of these different RM systems were spread through different species of bacteria through horizontal gene transfer, it just seems ironic that the system that is going to identify what is self and destroy what is non-self was at one point non-self. Okay, but maybe it's not as weird as it sounds. Maybe the dichotomy between self and non-self is what is weird. I mean, what does it mean to be yourself when more of the cells in and on your body are not you, but instead are bacteria and viruses, many of which create a symbiotic relationship with us that is both beneficial and necessary for our survival? And so on a larger scale, everything is tied in with this environment. We don't live in a vacuum, but rather are surrounded and influenced by those around us. Would you have the accomplishments that you have today if it wasn't for those who have helped you? What does it mean to be you without the support and influence of your friends, family, and those who have made you who you are today? And with that, see you next time. Oh, also make sure to check the links in the description so that you can read more about the research that was discussed today.